Hey everybody, my name's Katarzyna Komolibus and I'm from the Biophotonics group at Tyndall. And today I will talk about upconverting nanoparticles and certain challenges that they might help us tackle in different biophotonics applications. So this is our webinar 7 and you've been through other 6 webinars so far. So you got acquainted with different applications and different techniques that we use in biophotonics. So you're aware that there are certain limitations, especially where when we work with visible or ultraviolet light, because of the attenuation of different wavelengths, and that's caused by the absorption of different chromophores in tissue. We are working typically within the optical window, and also biological tissue is highly diffusive. So all of that together leads to poor penetration depth. So when we are working with, for example, treating deep-seated tumors, and we would like to look deep inside of the tissue, that's causing a lot of problems with the penetration depth and with the resolution. And also when we are using visible or ultraviolet light to excite the tissue, or fluorophores that we work with, we are also causing autofluorescence, and that's adding a background to our signal and reducing our signal to noise ratio. So this is where the upconverting nanoparticles come into play. And today mainly I will focus on kind of brief introduction to what upconverting nanoparticles are, and I'll try to walk you through a few applications depending on the time that we have left towards the end. So upconverting nanoparticles are inorganic crystalline nanomaterials, and their main feature is the fact that they can convert near-infrared light into visible, ultraviolet, or also other near-infrared emission light. So they are typically less than 100 nanometers in diameter, and you can see a TEM image of the upconverting nanoparticles in the right-hand side of the slide. And they were first demonstrated in about early 2000s. And since then, they really attracted a lot of attention from different research groups because they are very suitable for a lot of applications, and that's what we will talk about today. So upconverting nanoparticles consist from this inorganic host matrix, and that's typically oxides or fluorides, like for example, sodium yttrium fluoride. And that host matrix is doped with lanthanide ions, meaning thulium or, for example, erbium. And those lanthanide ions act as luminescent centers. So why we would like lanthanide ions to act as luminescent centers? Well, they have a very special optical properties. And if we look at the periodic table, the lanthanides are typically located kind of below the periodic table, and they contain 15 elements from the element 57 through to 71. And what's special about them is that they have at least one electron in the 4f orbital, or at least majority of them do. What's actually special about this is that this 4f orbital isn't the most outer orbital that they have. So before they start filling the 4f orbital, they actually fill other outer lying shells like 5s or 5p, for example. So if we look at the electron probability, where we can find electrons as a function of the distance from the nuclei for gadolinium, on the right-hand side of the slide, you can see that the 4f shell lies kind of the closest to the nuclei, and then the other shells, 5s, 5p, 5d, and 6s, follow afterwards. So they are kind of outside, even though that they are fully filled, while the 4f shell is still not fully filled. So those valence electrons found in 4f shell are actually kind of buried underneath the other fully filled shells. So that's leading to a lot of different properties. For example, very weak absorption, narrow emission lines, long intermediate excited state lifetimes, which can vary from microseconds to even several milliseconds. So when we look at the energy diagrams for the lanthanides, what we can see is that they have a very rich energy level structure. So this is because there are strong Coulomb interactions, so electron-electron interactions also spin orbit coupling, and to a bit lesser degree, crystal field. And all of that leads to this energy level splitting. And that's why you can see in the image at the bottom of the slide, the energy levels for various lanthanide ions. And you can see that they create this kind of ladder-like structure. So this gives the opportunities for electrons to kind of hop from one level to another, to a higher and a higher level, providing that there's absorption of the energy from incoming photon. So those 4F transitions are in theory forbidden, but because there is a mixture between mixture of different parities between different orbitals, they are actually present. So we can actually use them for emission. So because those energy levels span such a wide energy range, we can actually tune the emission and the absorption of lanthanides to wavelengths that suit our application. So by choosing different ions, we can select the emission wavelength. So for example, in biophotonic applications, what we really like is working with near infrared, because that's our optical window, and that's where we can penetrate deeper. So we would like to use ideally both excitation in near infrared and also emission in near infrared. So how does the upconversion emission actually works? So the upconversion process is a process where two or more sequential low energy photons are converted into one higher energy photon via intermediate state. So because we are converting lower energy into higher energy, we call it anti-Stokes shift. So in contrary to traditional fluorescent dyes, where we use higher energy to convert into lower energy, and that's called Stokes shift. So at this point, it's also important to notice that the lanthanides are not the only possibility of converting multiple photons, low energy photons into high energy photons. We can also observe it for example, multi-photon microscopy or second or third harmonic generation. But those techniques require short laser pulses and very high energy, which means that they might not be ideal for some of the biophotonic applications. So that's why the upconversion emission is so beneficial, because it doesn't actually require such a high power to generate it, and we can use it with biological tissue. So there are three different processes that can cause upconversion, three main processes. There are a few different kind of side processes, but we won't talk about them because that would take too much time. So the simplest one is the excited state absorption. The next one is energy transfer absorption and photon avalanche. So because the energy transfer absorption is the most commonly occurring upconversion process that we use in upconverting nanoparticles, I'm going to talk a little bit more in detail about that one. So how does it work? In energy transfer upconversion, we have two ion systems. So we have a sensitizer that absorbs light, and we have an activator that emits light. Okay, so we have two lanthanide ions, and providing that they are within close proximity from each other, once the sensitizer absorbs light, it can transfer that energy to the activator. So first requirement is that they are in close proximity, and the second requirement would be that their intermediate state energy levels are close in energy. Okay, so that these electrons can, so that the energy can be transferred from one ion to another. And once that happens, the activator is going to store that energy until the following photon comes in. The sensitizer can sense it and transfer it again, and then the activator has enough energy to actually emit light. So the two most common examples of pairs based on two lanthanide elements are erbium and ytterbium, and then thulium and ytterbium. So if you look at the image on the right-hand side of the slide, we can see the emission from both of them. So we excite them in near infrared. We excite them with 980 nanometers, so we can penetrate relatively deep with that wavelength. And then the ytterbium is used as the sensitizer. So that's the ion that absorbs the light, and thulium and erbium are used as activators, and they are the ones that emit light afterwards. So in the case of combination with thulium, we observe blue emission, and in the case of erbium, we observe green emission. But these are not the only lines that we can use with those ions, because if you remember the energy levels that I've shown you before, there were many other possibilities. So that depends on the synthesis and optimization of the nanoparticles. They can actually emit different wavelengths from the same ion. So if we look at the energy transfer upconversion process in a bit more detail in the animation on the left-hand side, I'm not sure if that's still playing. Okay, I think it should be playing right now. Maybe I'll go. Okay, one more time. So we have an incoming photon, the ytterbium, which is our sensitizer, absorbs the energy, transfers it to thulium, and promotes an electron to the first excited state. And then after subsequent absorption by ytterbium, it promotes the carrier within thulium from the first excited state to even higher state. And after that, this state is an emissive state, and it's going to emit 800 nanometers line. Okay, so we have, in this case of ytterbium and thulium, we can not only have the blue emission, but we can also have a near-infrared emission, which is actually ideal for biophotonics, because that means that our both excitation and emission is actually penetrating quite deep in tissue. So how efficient is that process? Well, not really efficient, and that's the issue. So that's the actual bottleneck of the upconverting nanoparticles. This upconversion process is not very efficient. So when we compare it to fluorescent dyes that are typically used in bioimaging applications, you can look at the quantum yield, which means the ratio of the emitted photons divided by the number of absorbed photons, and you can see that it's constant as a function of power density, and it's typically quite high. It can even reach 90%. While in the case of upconverting nanoparticles, which you can see on the right-hand side, it's not only very low, it's typically below 5%, of the order of maybe 2 to 3%. It's also nonlinear, because our process is built on multiple photons that have to be absorbed before we can get our emission to work. The quantum yield curve is also nonlinear. So the slope of our emission would be the slope of the emission, intensity of the emission would be related to the number of photons that take part in the upconversion process. So in the case of the thulium, where we had two photons absorbed by the ytterbium, as we saw on the previous slide, the slope would start at 2, and then it would slowly decrease towards 1 when the system is slowly reaching saturation. So what's the reason for this low efficiency of upconverting nanoparticles? Well, first of all, it's the weak absorption, and that comes from the fact that we have these 4F orbitals, that they are kind of buried underneath the other orbitals that I mentioned before. The other reason is that those very complex energy levels, they are beneficial for many reasons, because we can use them as the stepping ladder to achieve the upconversion, but also they create many pathways for non-radiative recombination. So that's detrimental to our emission. And the last point is that because upconverting nanoparticles are relatively small, they have a large surface to volume ratio, and typically surface states can cause a quenching effect. So they are detrimental also to our emission. So when we talk about efficient upconverting nanoparticles, what we would really like to see is optically transparent and low lattice phonon energy host matrix. We would like to see a high absorption cross-section sensitizer. We would like to see a suitable activator, and by suitable activator, I mean an activator that matches the intermediate state of your sensitizer, first of all, and then that it's optimized for emitting the wavelengths that we need for our application. And last but not least, we would like to we would like to avoid those surface effects, so we can actually grow a shell around those upconverting nanoparticles, and that will protect our particle from those quenching effects. So there are a few other methods that can be used to actually improve quantum yield. For example, multi-layer structure. So when we kind of stack up those shells, we call it a multi-shell structure. So then we can improve not only the absorption, but also the energy transfer between the ions, and that could improve the quantum yield. We could also surround our nanoparticles with absorption dyes. So absorption dyes typically have a larger absorption cross-section, and then that energy that's absorbed by the absorption dye can be transferred to our upconverting, to our lanthanide ions, and subsequently increase the quantum yield of the upconversion process. And the last method of improving the quantum yield could be also using pulse excitation. So it's been shown both experimentally and numerically that if we use short pulses with optimized length and duty cycle, we can actually enhance the quantum yield of nanoparticles, and that would help us reach deeper tissue. So now that we know how the nanoparticles work and what to do to improve their efficiency, it's also very important what we need to do afterwards to use them in biological applications. So typically the nanoparticles after synthesis, they are hydrophobic, so they are not very suitable for aqueous media, which means that they are not suitable for majority of biophotonics applications. So we would like to do something about that, and we can actually perform certain steps in order to modify the surface and do surface functionalization in order to disperse the particles, for example, in water. And afterwards, what we can also do is we can add certain functional groups to their surface and also antibodies, which means that at this point, when we have the nanoparticles that can be both dispersed in water and have these antibodies attached to them, they can be used for targeting certain cells. Or in the example that you can see on the slide, they target actually bacteria. So depending on our application, we can work on the surface chemistry so that it provides the best specificity for our application, and we can incorporate those nanoparticles into biological samples, and they will attach to the cell of interest. So now we'll go through a few examples of where we can use the upconverting nanoparticles and how they could be beneficial there. So there are many of the applications that upconverting nanoparticles could be used for example, photodynamic therapy, in vivo imaging, super resolution spectroscopy, biosensing, in vitro imaging, or even optogenetics. So I'm sure you've heard about most of those applications before from either Marcelo, Brian, Stefan, or Carl. So I will try to talk about a few of them right now. So first of all, we would like to use upconverting nanoparticles for deep tissue in vivo imaging. So in this particular case, you can see how the upconverting nanoparticles were modified and functionalized to attach selectively to certain brain tumor cells. So here we were working with glioblastoma brain tumor, and what was shown here is that if we inject the nanoparticles into the mice in vivo and observe the emission at certain intervals, we can notice how the nanoparticles are actually accumulating preferentially within the tumor. So if you look at the right-hand side image on the slide, we can see that the mice was actually injected with 2 different tumors, just as a comparison to see whether the accumulation would be specific. So on the left-hand side, it was the tumor of interest, and on the right-hand side, where the arrows are pointing, it was a different type of tumor. So then when we look at the upconversion emission, we can see that after 1 hour, upconverted nanoparticles were mostly accumulated in liver and spleen and a little bit within the tumor of interest. And then after 4 hours, the accumulation was actually increased within the tumor site and decreased within the liver and spleen. And then after 24 hours, we can see that the upconverted nanoparticles were still within the tumor, and to a lesser degree within the liver and spleen. So this is just one of the examples how we could use upconverted nanoparticles to, for example, monitor tumors or other cells or bacteria in vivo by modifying their surface and trying to tag the cells of interest. So typically for these kind of fluorescence applications, we would use also fluorescent dyes and quantum dots. And this table presents how the upconverted nanoparticles actually compare to those two fluorophores. So we mentioned that because we are exciting upconverted nanoparticles with near infrared and they are emitting in near infrared, we have increased penetration depth, which is not the case for the other fluorescent probes. And also because we are working with the anti-Stokes shift, we don't have really the autofluorescence. So apart from that, they are photostable, so they don't bleach, which is a good advantage over the fluorescent dyes, for example. They are considered non-toxic, although I have to admit that there's still research needed in that area to understand a little bit more how long they are preserved in tissue and what kind of long-term side effects they could cause. They also have very narrow emission lines, which is not the case for both quantum dots and fluorescent dyes. And that, for example, enables multiplexing. And apart from that, they can be functionalized, which is also very useful for all biological applications. So the only drawback really in comparison to other methods is the low quantum yield. So as we talked about it before, there are many different methods how we can try to improve the quantum yield. And as you could see from some of the applications, it actually works. So another application for upconverting nanoparticles would be immunohistochemistry. So what we can see here is actually upconverting nanoparticles that were surface modified to target HER2 receptors, which are very important receptors in diagnosing breast cancer. So depending on the receptors present in the cancer, the treatment, the following treatment would be different. So typically, the pathologist would use DAPI to observe whether there is any HER2 within the excised specimen, while in this case, we also had as a comparison the upconverted nanoparticles targeting the HER2. And what was shown is that they actually achieved 50-fold better signal-to-noise ratio than conventional fluorescent labeling. So going a little bit farther from that, we can actually do multiplex detection. So in this case, what we had was targeting multiple receptors. So ER, PR, and HER2, they are three very important breast cancer receptors. And what was shown in this paper here was that because of the very narrow emission lines of upconverting nanoparticles that don't overlap each other, we can actually simultaneously achieve detection of 3 different receptors from the same cancer lines and also excised tissue. So this was a very nice result, and it's very encouraging for the future of digital pathology because in comparison to traditional immunohistochemistry, it actually achieved very, very good results. So going further, this kind of imaging techniques allowing for also quantification of the amount of receptors present in the tissue combined with artificial intelligence could be a great help to pathologists in providing the diagnosis. So another application of upconverting nanoparticles could be a photodynamic therapy. So in this case, you're probably aware that in photodynamic therapy, what we have is a photosensitizer that accumulates preferentially in the tumor site, and then it's activated with light, and it creates singlet oxygen, which kills the tumor cells. So because majority of the photosensitizers are excited with visible light, the penetration is not so deep. So when we are treating deeper seated tumors, it's actually that the light cannot reach them. So we could use upconverting nanoparticles because they are excited in near infrared, and they can emit also the visible light to reach deeper seated tumors while exciting the photosensitizer at the same time. So another application of upconverting nanoparticles would be super resolution spectroscopy. And I'm a little bit conscious of the time, so I might skip this one, even though it's very, very interesting. And jump to my summary. So in general, upconverting nanoparticles are inorganic nanomaterials which are doped with lanthanide ions, and they provide us with great optical properties for achieving deeper light penetration and a lot of other benefits for biophotonics applications. For example, increasing the signal to noise ratio thanks to reduced autofluorescence. We can also work with quantitative imaging, and because of such an ease in which we can modify the surface chemistry, we can selectively target different cells. So they really do show promise for a lot of biophotonic applications. So thank you very much.